I hope all of okay, okay, you are here. Okay, so we have to start with our topic, which is on proteomics. So I'm just asking you the thing that are you able to see the presentation? This proteomics, are you able to see it? Please let me know only once, then we will start our session. We are already very late. Any one of you, if you are able to listen to me, please let me know. Are you able to see the presentation? Any one of you, please let me know. Okay. Thank you, Nikita. Thank you. So we will have to start the topic which is on the proteomics. Okay. So I'm just and also one more thing. I will came back in between on this YouTube, and then I will take your, you can say if you have some queries or something, then we will discuss all these things. So I'm just starting the presentation now, and then I will came back. Okay. Okay. So thank you all for the playing. Yes. So we have to start the today's topic, which is on proteomics. Okay, so what is proteomics, how it came into existence, and what is its mean, what are the things which are there in proteomics. We have to cover all these things. So the term proteome was first coined in 1994 by an Australian, fellow, a postdoc fellow whose name is Mark Wilkers. So this term, which is proteomics, it comes into existence in 1994. Proteome refers to the total set of proteins expressed in a given cell at a given time. So you all know about this, that we have a you can say cell in which we have nucleus. In the nucleus we have the DNA, and we have another cell organelle is also there. So proteome generally refers to total set of proteins expressed in a given cell at a given time. That means at a given time, suppose it is a stage of reproduction or some another activities. So at a given time, how many proteins or the total set of proteins which are expressing itself is known as proteome. So to study the dynamic protein products of the genome and their interaction. So in proteomics, basically we have to thoroughly study, or we have to know about the proteins, about the protein products, and even the interaction which they have with each other. So a large scale characterization and functional analysis of the proteins expressed by a genome. So in the genome, when we have the set of genes, you can say, in the genome, when we are able to understand the functioning of the proteins or the expression of the proteins, then it is known as proteomics. So in proteomics, what will happen? In proteomics, we are able to understand the interaction which is there within the proteins, the functions of these proteins, everything related to proteins we are able to study under proteomics. So when we are saying genomics, then it is the study of the DNA. Okay, or you can say the genes, the chromosomes. When we are saying transcriptomics, it is a study of the RNA, which is derived from the DNA by the process of transcription. And when we are saying proteomics, it is a study of the proteins, the functionality of the proteins, the interaction of the proteins, everything which is related to proteins. And obviously we get the proteins from the RNA by the process of translation. So we have these recent terms which are there. We have genomics, we have transcriptomics, we have proteomics, and yes, we have another term which is known as metabolomics. And this is about the metabolite or the functioning of the body. It is another term. So we have genomics when we are using, or when we are doing the study on the DNA at the DNA level, then we are doing it at the RNA level, and then proteomics when we are doing it at the protein level. So I hope you all will be able to know the proteomics, the concept of the proteomics, or the term proteomics. So proteome is defined the complete set of proteins expressed during cell's entire lifetime. So it is defined as the complete set of proteins. Proteomics is the study of proteome. It uses technologies ranging from genetic analysis to mass spectrometry. Yes, we have so many technologies which are there. We have so many techniques which are there. I even have another presentation on the techniques only. The techniques which are used in the proteomics section. So we have a variety of you can say techniques which are available. We have genetic level techniques, we have mass spectrophotometry which is solely for the protein analysis. So all these things are there. Proteomics assesses activities, modifications, localization and interaction of the protein. So when we are discussing the proteomics, then we have to understand the activities which is there by the proteins, their interactions within each other, their modification, their functionality. We have to discuss everything in the proteomics. So this is the thing when we are studying at the chromosomal level, it is genomics. When we are studying at the RNA level or DNA becomes RNA, okay, then it is transcriptomics, and proteomics is the totally when we have to study about the proteins, their functionality, their activity, everything, it comes under the proteomics. So these are the, you can say, branches in its own self. Genomics has so much in its own self. Similarly, RNA has so much in its own self. So these are the, you can say, different wild varieties, or you can say branches of the, when we are doing the functioning of DNA, RNA or protein. So these are the, but right now if you are at the today's era, at the today's time, this proteomics is quite important. So lots of researchers are there, lots of scientists are there who are working at the protein level. Okay, so this field is quite important these days, proteomics. So, proteome indicates the total proteins expressed by the genome in a cell or a tissue. So, when we are even when we are discussing the activity of a single cell, or when we are discussing the activity of multiple of cells, so when at even at a single cell, when we are discussing the activity of the proteins only. So, it indicates the total proteins expressed by a genome in a cell or a tissue. Biomarkers detections might allow identification of patients who would benefit from further evaluation. With the development of proteomic techniques, proteome analysis provides a fast, non-invasive diagnostic tool for patients with various diseases. Yes, this proteomics, or when we are working on the proteins, these things are quite helpful in the diagnostic purposes. To advent of highly sensitive proteomic technologies can identify proteins associated with the development of diseases well before any clinically identifiable alteration. MS has a high resolving power and identifies proteins with more accuracy. MS is mass spectrophotometry. So these are the techniques which are there in case of proteomics, or when we are studying the proteins. This is again the same thing, that from DNA we are able to get the RNA, and from RNA we are getting the proteins, and then we are interacting the, or we are discussing about the proteins only. So why proteomics? Now this is very important thing, that why we are doing proteomics, why we are focusing so much right now at the proteomics, why? So number one, the behavior of gene product is difficult or impossible to predict from gene sequence, because when we are working on at this step, they have some results which will came, like from DNA we will able to get the RNA, then from RNA we are getting the mRNA, but the only final results or the only final implementation will be done by these proteins only. Getting my point? So they are saying the behavior of gene products is difficult or impossible to predict from the gene sequence, because we are able, yes, they have their own rules also, but we are able to get the final product from the proteins only. So if a gene is transcribed, its expression may be regulated at the level of translation. So that is transcription, translation, they are quite related with each other. But when we are getting the proteins, then this is the From proteins, there is not any another gene sequence or any another thing after protein. So there is only proteolysis, that means the breakdown of the protein, or there is some post translation modification of the proteins. So from this end product we are able to get the result. Getting my point? So that is why proteomics is quite important these days. Are you able to get to me? Let me check it only once. So are you able to listen to me? Everything is fine? My voice is clear? My voice is clear to all of you? Everything is fine? Okay, okay. Thank you, Shalja. I'm just checking it whether you are getting it or not. Okay, okay. Thank you, Prateek. Thank you, all of you. So just we have done that why proteomics is important to all of us. Okay, okay, okay. Thank you all. Thank you. Okay. So yes, this is the that why proteomics is important to all of us, or why it is so much prominent these days, because this is the end product, and from here this is not any another thing which we will get. So this is the end product, and this is the quite important. Then we have types of proteomics. When we are discussing about the proteomics is based on three main types: structural proteomics, functional, and expression proteomics. Now what happens in structural proteomics? The ultimate aim of this proteomics is to build a body of structural information that will help predict the probable structure and potential function for almost any protein from knowledge of its coding sequence. So yes, when we are discussing about the structural proteomics, our main focus is on the structure of the protein. Okay, there are 3D structure, there are 2D structure, or you can say either primary structure, there is secondary structure, there is quaternary structure, tertiary structure. So our main focus is on the structural part. But when we are asking, or when we are saying about the functional proteomics, it refers to the use of proteomics technique to analyze the characteristics of the molecule in a living cell. So our main focus is on the, you can say our main focus is on the functionals of. So our main focus is on the functionality of those proteins, the function which that protein is covering, the activity of those proteins basically. Then we have expression proteomics. It refers to the quantitative study of protein expression between samples differing by some variables. So in expression proteomics, you all know that expression means when we are adding something, or when we are doing some type of chemical reactions, so we have to know the expression, or when we are adding something in the chemical, or in, you can say, in the solution, so there is some change in the color or something is like that. So it refers to the quantitative study of the protein expression. So these are the types of the proteomics, or you can say the study of the proteins which is there. So what is the objectives of this proteomics? How this proteomics, or how this branch is doing all the things? How? So yes, in this we have when we are doing proteomics, so first of all we have to do the protein peptide separation. Now what do you mean by protein peptide separation? That means we have to separate the types of proteins, the proteins which are present. We have to do the separation of these proteins by some techniques onwards. Then after the separation we have to do identification and characterization of the proteins by MS. MS means mass spectrophotometry. So the proteins which we get, the resolved proteins from this separation, we have to check it under the spectrophotometry. And then from this mass spectrophotometry, we are able to get some data, and from those data we are able to get the applications, or we are able to get the results which we have to implement further. This is the thing which we have already done before also. The structure, which is primary structure, which is tertiary structure, which is secondary structure, you all know about it, but at least let me cover it now. So the sequence of simple amino acids or sequence of specific amino acids in the polypeptide chain, it is a primary structure. We have glycine, phenyl and glutamine. All these amino acids, they are joined together in the primary structure. What happened in the secondary structure? The primary polypeptide chain of alpha helix or beta plated sheets. You already done this before, so we have alpha helix plated sheets. Then tertiary structure is what basically? The scan chemically to form three-dimensional shape of the protein chain. So like this is one polypeptide chain. So we have the interaction different polypeptide chains. So they are the different polypeptide chains like alpha chain and beta chain. So this is the quaternary structure, okay. Then after it, this is the workflow, this is the pattern of study, how to do the whole thing which is there in proteomics. So first of all, we have to do the protein sample collection and storage. Then we have to do the processing of these samples. The storage may be from samples we have collected, maybe from blood, from muscles, from anywhere. We have to do some separation by SBS phase, by 2D gel electrophoresis, by any of the methods. So this is the, like here we am already told you that we have to do the separation. So first of all is separation. Number two is the identification by mass spectrophotometry. So this is the thing. First of all, we have to do the separation, then we have to do it, the protein identification on mass spectrophotometry, and after it, after the protein identification, we have to do the bioinformatic analysis. Getting my point? Getting my point? Are you able to understand what I want to say? You have to start from the sample, or you have to take the protein part from it, and then you have to do the, you can say, you have to do the separation of the protein. When these proteins get separated by any of the techniques which are there in our lab, you have to identify the proteins on the mass spectrophotometry, or some other techniques are also there. After the protein gets identified that this is this protein, then there is a bioinformatics analysis. So this is the procedure of proteomics. I am just briefly telling you about what is proteomics. Right here I am not telling you the functions of the proteins. Proteomics, that how there is everything happens in the proteomics. So first will be separation of the proteins, and these will be separated by 2D gel electrophoresis, by HPCL also. Maybe some of you are there from the higher studies also, so you must have done these workshops, or maybe practically also they have done this. Then we have analysis of proteins, and at the last we have the database utilization. Okay, even when we are working on the phylogenetic software, even when we are doing the analysis on the DNA and RNA, at that time also everything we have to do, we have to separate the DNA or the RNA. Then we have to do the analysis of those sequences, and at the end, by doing the phylogenetic analysis or by using different softwares, we are able to get the data. So this data is quite important. This database is important for us. So these are the 5 steps of proteome analysis. First of all is sample collection, handling and storage. Like you are doing it on the blood sample, or on the muscles, on the hairs. It depends upon you, or according to your research on which sample you want to do the proteomics or proteome analysis. Then you have to the separation of the individual proteins by 2D electrophoresis. After it, protein characterization. The last step is very important, which is the storage of this data by using the bioinformatics software. So when we are doing bioinformatics, we have different softwares for the storage of not only the protein but also the DNA and RNA. So are you able to get it? Then when we are doing the proteomics, it is not only for proteins, it is also for the DNA and for the RNA also. Our main motive is to do the data storage. The data may be stored by you by using some software. The data is also available online also for your comparison of purposes. So in bioinformatics, the main point is the storage of data. But how you will be able to store the data? First you have to do the, you can say, the collection of the samples, and you have to do separation, identification, and at the last you have the storage of the data. So these are the proteomics techniques which are there for the separation of proteins. SDS phase is there, 2D gel electrophoresis is there. Maybe not maybe, but most of you must have done all these techniques. Okay. If someone is new, then please let me know. I will share some PPTs with all of you. So this is by gel based techniques. So in both these SDS and the 2D gel electrophoresis, we are using the gel, which may be agarose, which may be polyacrylamide. So we are using the gel. And then we have off gel base, when we are not using the gel, like we have amino acidity technique. Okay. And after it we have quantitative proteomics. These all are the methods for the separation of the protein. Like we have mass spectrometry, which is in which we have different types. This is also for the separation of the protein. So again, this is the flow diagram that how is the separation of the protein. It may be gel based, that when you are using it gel, 1D gel, 2D gel, SDS gel, or it may be some another method like digestion of the protein mixture and then separation of the peptides. But at the end you have to do the mass spectrophotometry to get the data, to do the protein identification, quantification, and for protein validation you have to do the mass spectrophotometry. Now these are the techniques of structural proteomics. So this is the general proteomics which we have done or which we have discussed till now. I hope you will be able to understand what is proteomics and what are the techniques or what are the steps of proteomics. So when you are doing proteomics, not only proteomics, when even you are doing genomics or transcriptomics, first of all you have to do the identification or you have to do the collection of the sample, and then you have to do the separation. After separation, then another step is the identification of the protein or the DNA sequence or the RNA sequence. When you are able to identify the sequence, then after it you have to do the data storage, which is the third step. So you have to remember all these things in your mind, that the number one is separation, number two is analysis, or you can say identification, and number three is database utilization. So these all is the general terms which are there, the broad spectrum of the proteomics. Then we have techniques of structural proteomics. So structural proteomics is basically what? When we are focusing on the structure of the proteins. So the large scale analysis of protein structural comparison can help identify the function of newly discovered genes. So we have X-ray crystallography, we have NMR spectroscopy. By all these we are able to understand or we are able to know the structure of the proteins, and which is quite helpful to identify the function of newly discovered gene. Suppose if there is some gene which is discovered, which is not discovered before, which is discovered now. So by using these structural techniques, we are able to know the functioning of that particular gene. So we have X-ray crystallography, we have NMR spectroscopy. So as you all know that these crystallography and spectroscopy, they are the part of physics or chemistry basically. So in bioinformatics, on the very first day I told you all that bioinformatics is a combination of all the branches of the science. So these are the protein separation techniques. We have SDS phase, we have 1D, one-dimensional SDS phase, and we have 2D, which is two-dimensional SDS phase. I hope you all have done SDS phase, so I will not do it now. Basically what happened in this SDS phase, we have the separation of the proteins based on their charge, the pH which is there. So they will separate like this. The small one is there, the larger ones are there. So they will separate according to their charges. This is 2D gel electrophoresis, which is for the separation and identification of proteins in a sample by displacement in 2 dimensions. That is why it is known as 2D gel electrophoresis. So this is also the same thing. We have to do it on a gel, and then we are able to get the band. Okay, and this is the actual gel which we get, the actual result which we get by using the 2D gel. So approximately 1000 E. coli proteins on a single 2D gel. So here we have the presence of, like, these dots are the proteins. These dots, these are the dots here now. All these, so approximately 1000 E. coli proteins are separated here by using 2D gel electrophoresis. So this is electrophoresis, then after it we have HPLC. Let me check whether you are here or not. So are you getting it? Everything is fine till now? Please let me know. Everything is fine. Please let me know. I hope it's very easy. Everything is very clear till now. Please let me know everything is okay. Harshdeep, Prateek, everything is clear, students. Aradhana, Jerry, Soumya, everything is clear. Bache, please let me know. Okay, okay. Thank you, Shreya. Okay, 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 Geetika, okay. So I hope it's very easy thing. We are not discussing anything difficult till now. Everything is very easy. But you have to know the basic, the fundamental thing which is there in the proteomics. The processing of the everything which is there in proteomics, we have to know. Okay? Okay, okay, okay. So now we will just move forward or move further, not forward, but it should be move further. So yes, we have done the electrophoresis, we have done the methods by electrophoresis, how we are able to separate. Okay, now we have another technique which is known as HPLC. So we have another technique which is known as HPLC. Basically it is a chromatography, and this technique is also quite used when some students are pursuing their PhD or maybe in their MPhil or MSc also. They are using the HPLC, which is basically the chromatography, and we are able to separate the protein. which are then processed further to the mass spectrophotometry. So what is mass spectrophotometry? Or you can say why mass spectrophotometry is so much important for protein identification. How and why? So the very first line is mass spectrometry is used for protein identification. That this is this protein. Identification, what do you mean by identification? Like we have so many students, we have Aman here, we have Jennifer here, all of you. So we are, how I am able to identify you? Maybe by your names, maybe by your looks, by your appearance. So mass spectrometry is used for protein identification. It is useful to obtain structural information like peptide mass sequence. So, by using mass spectrophotometry, we are able to know the structure of the protein. It is also useful in identifying type and location of protein modification. So, if there is any change in the structure or if there is any modification in the structure, we are also able to identify that thing also. So, a mass spectrometer separates proteins according to their mass to charge ratio. We have to remember this thing that in mass spectrophotometry or mass spectrometry, the separation is based on the mass to charge ratio. That means how much molecular weight is there, the charge which the protein is carrying. According to it, we are able to get the, or we are able to separate the protein. Getting my point? So, in mass spectrometers, proteins are definitely separated, but according to their mass to charge ratio. The molecule is first ionized. The process of ionization of protein forces them to move towards the analyzer because of the charge of the ion. So, they are just identified on the basis of their charge, on the basis of their molecular weight, on the basis of their mass. So, this mass spectrometry definitely provides us the molecular weight which the protein is carrying and their structural information. Getting my point, the structure of the protein. If there is any change in the structure of the protein, we are able to get from this mass spectrometry. So it always works with positive ions. So as we already discussed that we have the analyzer in the, you can see the software is there which will attract positive ions towards itself. So that is why this mass spectrophotometry or mass spectrometry is based on the mass to charge ratio, number one. Number two is we are able to know the weight of the protein. That means how many kilodalton the weight of the protein is there, and obviously we are able to get the structural information about the protein. So this is the mass spectrometry. So like in 2D gel electrophoresis, proteins get separated. It does not identify them. Even we have seen there is a separation of the protein. The dots are there. There is a separation of the protein. Here. But we are not able to, you can say, we are not able to know the molecular weight or something like this. But here we are able to identify the protein. So MS is used to identify them which separate charged particles or ions according to their mass. So you have to remember this thing that this is mass spectrometry. Here the separation is based on the mass. According to their molecular weight there is a separation. So two types of mass spectrometer instruments are there which are used in the lab, which are used in highly specialized labs. This is the function of this mass spectrometry. I will not go in the detail of this mass spectrophotometry or spectrometry basically. we have to remember only one thing, that this mass spectrometry helps us in the separation or in the identification of the protein on the base of the mass which the protein is carrying. So these are the different components of mass spectrometer. We have not to go that much in detail. We have source, we have mass analyzer, and then at the last we have the detector. From the detector we are able to detect the results. Yes. Then we have another important thing which is known as protein fingerprinting. You must have listened to the term which is DNA fingerprinting. Okay, all of you. But what is protein fingerprinting? So protein fingerprinting, also called peptide mass fingerprinting or peptide mapping. It is a technique for the identification of proteins. Like when we are using DNA fingerprinting, what we are doing? Our main focus is on the DNA. Okay, like there is some crime, there is some murder or something is that we are just focusing to collect the sample, and we are just focusing on the DNA because everyone DNA is unique. But when we are doing protein fingerprinting, our main focus is on the identification of the protein. So separated protein spots are obtained from the gel and then identified using protein fingerprinting. The method is based on the use of a proteolytic enzyme to digest the protein into a number of smaller peptides. So suppose this is a protein, suppose this is a highly compact protein. So we are using the enzyme so that we will be able to break this protein into the polypeptides. Okay, so unknown protein of interest cleaved into peptide by protease. The most commonly used protease is trypsin. All of you must know about trypsin, which cuts the protein at the lysine and arginine position. So trypsin, what it will do? It cuts the protein. So when the digestion is complete, a set of peptides are produced of varying masses that are unique of that protein. So when we are using this protein fingerprinting, or when we are using the trypsin enzyme or another set of enzymes, we are able to get the set of peptides, different peptides. So this is peptide mass fingerprint, or also known as PMF. In it, what will happen? Suppose this is the proteins, different proteins. The size is different. We have to, what we have to do? We have to add the digestion enzyme, which is basically the trypsin. When we are using the enzyme, what will happen? We are able to break the protein into small polypeptides. Then we have another term which is known as protein microarrays. Now what is protein microarrays? This is another strategy for large scale study of proteins similar to DNA microarray. Like we have DNA fingerprinting, similarly we have DNA microarray. Now what will happen in DNA microarray? What will happen in protein microarray basically? A very small amount of different purified proteins are placed on a glass slide in a pattern of rows and columns. So what we are doing in this microarray, what will happen here? We are using small amount of purified proteins which are placed on a glass slide in pattern of rows and columns. So we are just putting here on this glass slide. So this is a slide, and just we are putting it in a manner of rows and columns. So they have small points in which we are adding our protein. Followed addition of various types of probe molecules that are fluorescent dye labeled to the array. So now what we are doing? We are just introducing the, you can say, the probes. We are just introducing the probes here. What will happen after it? When the probes bind to the label, it results in fluorescent signal that can be read by the laser scanner. So when the probe binds to the label, it results in fluorescent signal. When the probe, which carries a fluorescent thing, when it binds with its specific target, what will happen? We are able to get the color. Thus, this technique can detect thousands of proteins interaction, can screen the ability of protein to bind other protein in complex, receptors, antibodies, lipids, enzymes, hormones, specific DNA sequences or small molecules, even new drugs. So when we have specific probe for specific thing, like we have specific receptors for which we have the probes, we have specific antibodies, like on the 2D gel electrophoresis we have thousands of protein molecules, but we have specific probe for only 10. For only 10 protein molecules, the probe will bind with only 10 of those. The rest will be wasted. So they have, if we want to do something related to enzymes, we have specific probes for those enzymes. So with this technique, we are able to get the specific reaction. So you will able to get the, when you are using the probe, it will bind to the specific receptors which is there. Then we have protein-protein interaction which is there. So first, let me revise all these things. We have microarrays, we have fingerprinting, we have mass spectrometry. All these things are for the separation of the proteins plus identification of the proteins. Like we have gel electrophoresis for the separation, but we have mass spectrometry, we have protein fingerprinting, we have microarrays for the identification of the specific protein. Getting my point? When we want to work, we have thousands of proteins which we get by this electrophoresis. We have thousands of proteins, but our main focus is on only 10 proteins. So for the identification of those 10 proteins, we have mass spectrometry, we have DNA protein fingerprinting, we have protein microarrays. So I hope you will be able to get till now. So now we have to study the protein-protein interaction. So are you able to get it? Everything is okay till now? So for the protein separation, we have electrophoresis, we have 2D gel electrophoresis, we have 1D gel electrophoresis. But for the identification of a specific protein, suppose we want to do something on a specific protein, we have spectrophotometry, we have this protein fingerprinting, we have protein microarray. All these things are for the protein separation plus protein identification. Everything is clear till now? Okay. Thank you, Meghna. Thank you, Prateek. So I am not taking very difficult part today. We are just doing it very slowly and we are just doing very simply so that you will able to know the basic concepts of proteomics. What is basically proteomics? What we are doing in the proteomics? What are the techniques which are there in proteomics? Okay. So now we have another term which is protein-protein interaction. Yes, this is another term. Now, what is protein-protein interaction? We all know that this type of interaction will occur only when two or more proteins bind together. So, this is protein 1, which is present on the cell membrane, or you can say on the outside of the cell. Then we have protein 2, which is inside the cell. Protein 3, protein 4, protein 5, 6 and 7. So, they are just interacting with each other, and they are also, you can say, passing some signals from one protein to the two, from second to the third. They are passing some signals also. Yes, this is another, this is, you can say, one system, or which we are describing the protein-protein interaction, that how the protein 1 is interacting with the protein 2. Before it we have done the techniques which are there in the proteomics. Now we have protein-protein interaction. So this is the yeast to hybrid system for protein-protein interaction. So most of the yeast to hybrid system utilize the reconstitution of an active transcription factor to assay for protein-protein to make the interactions. So what happens in this system? In this system, they are predicting the interaction between the proteins. The system requires two yeast hybrids be prepared called bait-prey system. So basically we have two yeast hybrids. The bait protein is fused to a transcription factor DNA binding domain and the other prey protein is fused to a transcription factor activation domain. Let me show you a diagram if we have. Yes. So we have two hybrid systems. One is known as bait and other is known as prey. So yes, we have one is the DNA binding domain and another is known as the transcriptional activation domain. So these two proteins, they will obviously they will interact with each other. Like this protein has specific receptor for this binding partner. So they will fuse with each other. What will happen then? When expressed in a yeast cell containing the appropriate reporter gene, interaction of the bait with the prey brings the DNA binding domain and the activation domain into a close proximity. So what basically happening here? They will come, you can say, very close with each other, and there is some interaction between there is some interaction between these proteins. Sorry. So, the bait-prey nomenclature has applied to in vitro method used to study the protein analysis. So, in vitro method for protein interaction analysis are often employed to confirm interaction indicated by this Y2H method, which is yeast to hybrid system for protein-protein interaction. So, when now they are interacting with each other, the two proteins, what will happen now? As you are able to see here, transcriptional activator binding site, it will get activated. Transcriptional activator binding site gets activated, and what will happen then? Transcription of the reporter gene, and this reporter gene will get the there is a transcription of the reporter gene, or we are able to get the reporter protein. By the interaction of these proteins, we are able to get the reporter protein. How this reporter protein becomes? It becomes by the activation of the reporter gene. So these all are the simple things which are there in the protein-protein interaction, or how we are doing the proteomics. Okay. Now these are another terms which are there in the proteomics, the databases. What do you mean by databases? Like in the DNA also, I already told you the sequences are there. The DNA sequences, they are stored somewhere on some softwares. Similarly here we have some softwares. Like for data analysis, we have Sequest, we have Mascot, we have Peaks. All these are the data analysis search engines where we are able to get the data. And if we want to do the protein databases, if we are solely working on the proteins, we have NCBS. Like for the uh, DNA, we have NCBI. Similarly, we have NCBS. We have Swisscode. All these are the softwares, or you can say all these are the software, you can say. These are available on which we are able to get the protein sequences from. Basically, they store the data related to the proteins. So these are known as databases. Then another thing is applications of proteomics. Why we are thinking so much about the proteins? What is the need of doing proteomics? So first of all, we are able to identify the protein We already discussed it so much time that we are able to identify the protein, we are able to separate the protein, and after separation we are able to identify it, and then we are able to analyze it. Protein sample purity determination, we will able to know whether the protein sample is purified or not. Detection of post translation modification, yes, this is the important thing, that after protein the only thing which is left is post translational modifications. So with the help of proteomics we are able to know the these modifications which are there after the protein part. Even we are able to detect the amino acid substitutions. Mass fingerprint identification of protein is also there. We are able to do the nutrition research, like that this protein is good for us, this is good for our health or something like that. To identify unknown protein of interest, quantify the protein and peptide, protein biomarkers. So these all are the applications of the proteomics. Definitely everything will be covering about the proteins only. So this is the conclusion of proteomics. Proteomics is a composed study of set of proteins. Number one, we have so much different types of proteins. So it is a set of proteins. The detailed protein studies will shed light on the role of protein modification in the protein function, that we are able to know the structure of the protein. So if there is modification in the structure of the protein, how the functionality will become different. The development of proteomics renders us with a powerful tool to examine biochemical processes at the molecular level and identify set of proteins. So you have to remember this point also, that not only the separation of protein is needed, the identification of the protein is the most important thing. The identification of protein is the most important thing. So during plant life, sometimes in adverse conditions, it can passes through biotic abiotic stress. At a proteomics can be useful to identify set of proteins. So identification of proteins at a molecular level, even during adverse conditions, we have to know the functionality or the structurality of that particular protein which is involving in the adverse condition, which is there during the adverse condition. So this is the proteomics part. I hope you are able to get it. This is again the same term, genome. We all know that sum total of all the genes. Genomics, the sequence when we are studying the genes. Transcriptome when we are studying the RNA. So I will not cover it again. Same things are there. So proteomics is basically what, when we are doing everything about the protein, it becomes the proteomics. So proteomics is the qualitative and quantitative comparison of proteomes under different conditions to unreveal biological processes is known as proteomics. When we are doing the qualitative and quantitative comparison. Okay, so again it is a science which concerned with the analysis of proteins. This is again genome. From DNA we are getting RNA, RNA to protein by the process of proteomics. So again, why do we need proteomics? You will just read it by yourself. Everything is very easy. Yes, this is very important point. Level of transcription of a gene, that means from DNA to RNA, is not equal to level of expression of a gene. Transcription is there. From DNA you are getting the RNA, but from RNA when you are able to get the proteins or the expression of a gene, sometimes it is not the same. So that is why the level of transcription of a gene is not same with the level of expression of the gene. Please keep this point in mind. Okay. So post translation modification is there. You can do it by yourself. All obviously we know about these structures, primary structures. So these steps are quite important. Protein separation, protein detection, or you can say protein detection by some software or some techniques. Protein analysis is there, in which we have to do the identification and in which we have to do the measurement of activity. So protein separation is by electrophoresis, that means 2D gel electrophoresis, by HPLC, by dialysis, by centrifugation, by salting out method. All these methods are for the separation of the protein. So you have to keep these all things in mind. Then we have to do the comparison. So this is the result. We have to do the comparison. Then mass spectrophotometry, again it is the mass to charge ratio. It is the separation of the proteins according to the mass to charge ratio. Getting my point? So ionization of proteins, ions propelled towards the analyzer by electric field. So we have ionization, we have detectors. These are the types of mass spectrometry. So mass spectrometry helps in the separation of different proteins. So by using mass spectrometry, like we have phosphoglutamase, we have HSP90. So we are able to separate the proteins. Then for protein analysis, we have crystallography, we have NMR. All these things are there for protein analysis. This is X-ray crystallography. Then we have NMR spectroscopy, which is a part of the chemistry basically. Then we have post-translational analysis also. This microarray we already discussed. Protein microarrays. After it we have the bioinformatics. In bioinformatics, like I already told you before, it is a building and manipulation of biological databases, integration of mathematical statistical, yes, integration of mathematical, statistical and computer methods to analyze the biological, chemical and biophysical data. So we have GenBank, we have AMBL for DNA sequences, and for protein sequences we have Swiss-prot and Flybase when we are working on the proteins. When we are working on the DNA sequences, we have GenBank and we have AMBL. But when we are working on the proteins, we have Swiss-prot and we have Flybase. So this is the thing we have already covered. These structures were already done before. This is some difference between genomics and proteomics. I will share the PPT with all of you. So you just read it by your own self, okay? So you have to read it by your own self. So this is a protein mixture. We have to do the separation. We have to do the identification with the help of MS. From the mass spectrometer, we are able to identify the proteins. So this is again electrophoresis. MS is there. This is mass spectrometer. Then fingerprinting we already done it, okay. Then microarrays we also done it. So you have this PPT along with you. So you just read it once. If you are not able to get anything, please let me know. Yes, this is another thing. Proteomics and drug discovery. Proteomics and drug discovery. This drug discovery or drugs are there. Some part of drug discovery is there in your topic. So, how this proteomic is helpful in the discovery of new drugs? How? Basically, by the data analysis, the data which we will get from that data analysis, we are able to get the new drug. I have to discuss everything regarding it. So we have some, you can say, certain protein implicated in the disease is identified by using the proteomics. 3D structure of a protein is generated using the software. Computer programs design the drugs to interfere with the actions of a protein, that is, a molecule that fits the active site of enzyme and cannot be released with the inactivate protein. So what basically helps, what basically is there, we have the protein molecule and then we are introducing some drugs on that specific protein molecule. Then we will come to know what is the effect of that particular drug on that particular protein molecule. So, computer program designed the drug to interfere with the action of the protein. So, it is the interaction between the drug and the protein molecule. A good example of this is identification of the drug to the target and inactivate the HIV-1 protease. So we have the protein which is HIV-1 protease. The virus cannot survive without this enzyme. So the virus which is there, which is HIV virus 4, it will not survive without this enzyme which is HIV-1 protease. Therefore, it is the most effective protein target for killing the HIV. So, are you getting my point? If we are able to discover, if we are able to discover that type of drug which will kill the HIV-1 protease. If HIV-1 protease will not be there, the activity of the HIV virus will become vanished. So that is why we have to target those drugs which inactivate the HIV-1 protease. So the virus will not be able to survive without this protease. So the drugs will work on a specific protease, or you can say specific protein on which they have to inhibit the activity of HIV-1 protease. Getting my point? So the drugs, these proteins, the proteomics is quite helpful in the drug discovery also. Drug discovery means when we are focusing on the new drugs. So it is the interaction between the drugs and the proteins. Then these are the applications of proteins. You just read it by yourself. Yes, these proteomics are there in the field of medicine, in the field of biotechnology, in the field of food sciences, agriculture. Everywhere the proteomics is participating. They have some unique role in every field. Okay, these are applicable in the field of medicine. They are helpful in the development and the aging processes. They are helpful in the drug discovery, they are helpful in the toxicology, they are helpful like their identification of novel drug targets, okay, surrogate markers, everywhere these are quite helpful to all of us. Getting my point? So proteomics is quite helpful in the field of medicine, in the field of toxicology, everywhere. Like this is diabetes at the molecular level. At this also, this proteomics approach or the proteins approach is quite helpful. Like changes that occur during healing. So they are telling us everything that how these proteins are helpful in getting or tell us about the results which are there by because of these proteins. So I hope you are getting my point. So these are the functions of these proteomics. I will not discuss it right now because you will be able to read it by your own self. I will share the PPT and you have to know what is microbial proteomics, what are the recent advances in the proteomics field. Okay. This is the little bit history of the proteomics and how proteomics come into existence. And this is the thing which we already discussed, that Mark Wilson is the one who introduced the term proteome. So you have to remember this thing, Mark Wilson. So these are the two scientists, which is Koichi Tanaka and John V. Fan, which got the Nobel Prize in 2002 in chemistry. For what purpose? Basically for the mass spectrometer for macromolecules for the development of soft desorption ionization methods for the MS analysis of biological macromolecules. So their main work is basically on the. Are you here? Are you able to listen to me? Everything is fine? Please let me know everything is fine. Please let me know. Fine. Are you feeling bored or everything is okay? Everything is okay? Everything is okay? Please let me know. With all of you, you will have to just go through the PPT I think everything is very simple. If you are not able to get anything, then discuss it again. So I'm just waiting for your answers so that I will carry it further. Megha, Soumya, Prateek, everything is fine. Please let me know. Okay, okay, Upasna. Okay, Megha. So, I just want to complete this topic. I hope you will able to understand it. Everything is fine to you. So I will share the PPT. If something is not clear to you, then definitely we will discuss it again. Okay, no issues at all. So just I'm just going to the PPT so that the proteomics will be clear to you. Even if you are doing your bachelor's, if you are doing your master's, or even if you are doing your PhD, then you will be able to tell anyone what is proteomics. Okay, so you have to be that much confident after this presentation that you will be able to tell anyone which is junior to you, which is your classmate or your colleague or anyone, that what is proteomics, what are the things which are there in proteomics. Okay, so I hope you are able to get my point. I have put so much in, you can say, so much time or so much presentation on proteomics only because proteomics is a very recent field. Getting my point? Proteomics is quite used these days. Even for the research purposes, if you want to do masters, if you want to do PhD, this field is quite important. So that is why I am saying to all of you that please, all of you have to study the proteomics part very briefly. Okay, okay, Priya. No issues at all, Priya. Don't worry. I am able to get your messages. Maybe little bit late, but you have to focus on the proteomics. You have to that much ability that someone is asking you about proteomics, you will have the answer about it. And even if someone want to know or some have some doubts regarding proteomics, you will be able to answer all those doubts. I will share the PPT. You have to go through this PPT. You have to revise it so that your everything will become clear. Getting my point? Manish, are you here? Manish, are you able to understand, student? Manish, are you here? If you are here, please reply. Everything is clear to you also. Please let me know, Manish, and if anyone else is there also, please let me know. Everything is clear to you. Please let me know. All of you, not only those two, but all of you, everything is clear to you. Please let me know. If you have some queries also, then please let me know. Otherwise, we will just complete it now. I'm not getting your messages. Maybe there is some. So, are you able to get it? Everything is clear to you. Please reply. Meghna is there. Soumya, Upasna, please reply. Soumya, Harshdeep, please reply. Prateek, all of you, please reply if you are able to listen to me. Okay, okay. Priya Shiri is there. Priya Shiri is saying, okay, ma'am. Okay. Okay, now just I am just completing the presentation. Just give me some more time. Yes. So these are the two scientists, which is Koichi Tanaka and John B. Pham, who got the Nobel Prize on this mass spectrometry for macromolecules. So this is the proteomics objective, peptide separation, or you can say protein separation, and then identification, characterization of the proteins by mass spectrophotometry and data analysis. We already discussed it, so I will not do. So this is again the softwares which are used for the proteins. We have sequest, we have mascot, we have peak, and we have protein databases which are NCBS, and we have Swiss spot. Even if you are interested in doing something related to proteins, so you have to type this thing Swiss spot on Google, and you will be able to, when you are able to click on the Swiss spot, you will be able to get the data or lots of data which is present on the Swiss spot, or even on the fly bank also. So if you are interested, just type these words on Google. Okay, any of these words, you will get lots of new things related to proteins. So this is again protein separation by SDS. I will not going to discuss it. You already know about it. The SDS gel electrophoresis, 2D gel electrophoresis, where there is separation of proteins on the basis of charges. Okay, this is amino affinity. This is also for the separation of proteins. Like if you are able to see here, we have mixture of protein, blue one and green one. But after this, we are able to get the blue one only. So we are able to separate the proteins. Mass spectrometry is there. Importance of protein, that they maintain the metabolic processes. They serve as structural elements both within and outside the cell. All these things you already know it. So this is basically sample collection. After sample collection, we have protein extraction, protein analysis, bioinformatics analysis, and this is the proteomic discovery. This is how the things are there in case of proteomics, how we are able to do identification, validation, functional study is there, then diagnostic and therapeutics use. All these things are there by the proteomics section basically. Then we have structural proteomics, where we are able to do structure. Computational biology is there. So we have the big perspective in the case of proteomics. We have mass spectrophotometer, we have gel electrophoresis, everything is there. Then proteomic application in diabetes, that how it is helpful in diabetes diagnosis. You just read it by yourself. Then proteomic applications in cancer, that how this proteomic application is quite helpful in the cancer, or you can say in the field of medicine. It is also useful for all these diseases, for pancreatic disease, for eye disease, for cardiac disease, for smoking, for everything. Everywhere we need the proteins, or you can say for every of these medicinal purposes, we have proteomics. Then proteins classes for analysis and characterization. Like we have membrane proteins, we have soluble proteins, we have nuclear proteins, we have so many different types of proteins. So we have to analyze all those proteins, and we have to do the separation of all those proteins. So this is again the same thing. We have structural proteins, we have expression proteins. These are techniques which are there. 2D gel electrophoresis is there, and then we have mass spectrophotometer. I will not discuss these things. These are some applications, like they have this is the important thing, that they are quite useful in the drug discovery. We already discussed with the example of IV1 protease. So like they are again giving us the example. Protease inhibitor is designed to disable the protease enzyme that allows particular virus to reproduce. That's HIV virus basically. The biomarkers is there. For tumor metastasis, we are using it. In neurology, we are using it. So I hope with all these things, this is all about the proteomics. Everything is same, but you have to read it, okay? Then you will be able to get it. Otherwise, it becomes difficult for you also. I think that's all. Uh, this is the like limitations of the proteomics. So you will just so you have to just go with all these slides. You have to you can say read it by your own self. If you will be able to get any problems, then please let me know. Otherwise, this thing is the proteomics will be definitely clear to all of you. So are you able to get it or not? Everything is still clear to you or not? Please let me know everything is clear. I hope it will be clear to all of you. It's a very easy thing. We have not discussed anything difficult till now. So yes, now I will share the topic, the syllabus which you have. So you have proteomics, then after it you have introduction to drug discovery. Are you able to listen to me? So we have introduction to drug discovery. So yes, from the proteomics onward we have the drug discovery part. But this drug discovery is from the proteomics section basically. So I have started the protein classification structure you have already done before. Protein structure prediction is there, proteomics we have done, and even we have touched something about introduction to drug discovery. We have touched some part of drug discovery. After it we have drug discovery technologies and strategies. So from tomorrow onwards, that is from tomorrow, we will do drug discovery and the technologies which are there in the drug discovery and the strategies which are there in the drug discovery. So if you are able to get the proteomics section, then you will be able to get the drug discovery section also, because drug discovery is quite related with the proteomics section. That is why I have started the proteomics today. It is very important thing which we have done today. From the proteomics we will tomorrow we will do the drug discovery. So we will start with the drug discovery tomorrow. Even we will try to complete the small things which are there in the drug discovery session. There are not small things, there are very big important topics which are there, but we have to start with technologies and strategies which are there in the drug discovery. Number two, any type of cell activity or cell cycle if there is present in case of drug discovery. Then another small topics are there like computer aided drug designs. Like even in today's presentation we have discussed that there is a computer aided drug designs are there which bind with the specific protein molecules and they change the activity of that particular protein molecule. Okay, so we will discuss that part tomorrow. Okay, after the tomorrow session, when we will be able to complete our drugs part, then we will move towards our phylogenetic part. Getting my point? So today we have covered proteomics. Tomorrow we will cover the drug discovery part or the drugs part, in which we have to do the strategies which are there in the drug discovery, the designs of the drugs, and after it we have to do the phylogenetic, which is your topic number third or fourth maybe. Introduction to phylogenetics and methods of phylogenetic analysis. So are you able to get my point what I want to say? Yes, yes, I will share the PPT. No issues at all. Please don't say this is my duty to share the PPT. Don't worry regarding it. But you have to tell me only one thing, that are you able to get what I want to say to all of you, that today we have done the proteomics part, tomorrow we will do the drug discovery part, and when this drug discovery part will become complete, then we will do the phylogenetic part, which is maybe your third or fourth topic. Getting my point? Are you able to get my point? Please let me know. I know the messages are coming a little bit late, but please try to reply if you are able to understand my point. Any one of you, please reply. I think this today's lecture will definitely be helpful for all of you in your future perspectives also. So please, I will share the PPT in the group also. So you have to revise it one or two times, and then everything will be clear to all of you. Yes, yes, Shalja. Thank you, Shalja. Ma'am, can we switch to interactive platform? Yes, Geetika, definitely we will. No issues at all. Meghna saying no, ma'am, please repeat. What to repeat? I'm not getting it. What do I have to repeat? I will repeat everything if you want to, but please let me know. I'm not getting your the message what you want to say. So, after the drug discovery part, we will go with the. Okay, okay, Geetika, definitely we will do. No issues at all. So after the drug discovery part, we will go with the phylogenetic part, which is maybe your topic number third or fourth, maybe. Yes, I think that's all for today's session. I hope this will be useful for all of you. And yes, with all these positive vibes and positive energy, we have to complete our 10-day lecture series. As I told you before, also I am there always for all of you, no issues at all, no matter what at all. It's my suggestion to all of you that please be focused on your study, or please be focused on the topics which we have to cover. You have to revise it two or three times, otherwise you will forget everything, okay? So you have to revise, you have to keep revise. So that's all from my side. Please take care of yourself. Tomorrow we will meet with drug discovery part. Maybe because of the signals, I am not able to get your replies. But definitely we will meet with the drug discovery part. If you want to say something, then please let me know. If you want to say something, please let me know. I'm not getting anything. That's all from my side. Please take care of yourself. We will meet tomorrow sharply at 8 p.m. Till then, take care of yourself. Bye bye. Good night. Because I'm not getting any messages, I'm not able to know what to say. Let me refresh it. Maybe now I'm getting. Yes, yes. Okay, bye bye, bye bye to all. Yes, I came to know your. Okay, okay, bye bye to all. We will meet tomorrow. Till then, take care of yourself. Okay. So just please take care of yourself. Now I'm just